right. So yeah, it's a great pleasure to be here. I'm not following exactly the template that was provided with all the exact slides that I have to talk about myself and about my projects and so on. But I would like to a little bit give my background also. So as you can hear, I'm from Germany, did my residency or my medical school in Erlangen and Freiburg in Germany. But actually what really got me into the field of Alzheimer's disease, but also into San Diego and in general also into UCSD was when I was doing a scholarship that I got from the German Research Society to actually do research here at UCSD back more than 20 years ago. That was during my medical school time. And back then I had no clue about Alzheimer's disease. I know it was age-related disease and so on, and I never heard about Leonthal before. So I was very ignorant at that time, and I didn't know there was back then talking to one of the greatest in Alzheimer's disease research and also in clinical trials. And I was very fortunate that he took me as a German medical student into his lab together with under the supervision of Don Piso. And back then I was able to work on nerve growth factor. So back then, more than 20 years ago, there were ideas and still are here at UCSD. So if you infuse growth factors, that may actually be beneficial, that you rescue neurons, and then you can slow down memory decline and so on. And I was very fortunate to work with him on that project. And one side effect that this nerve growth factor infusion therapy back then had in patients that got it was lower back pain. And we were always wondering why does it cause lower back pain when you infuse nerve growth factor directly into the brain, into the CSF? And so my work back then was to look at the peripheral nervous system, dorsal root ganglia. Dorsal root ganglia, they harbor the sensory neurons, and they're very important to transmit pain signals back to the brain. And what we could see or find out as the cause for the lower back pain that patients had when they got nerve growth factor infusions was that there was a proliferation of so-called Schwann cells, or cells that are around myelinating fibers. And you can see here in control rats and then rats that got nerve growth factor that you see a massive proliferation of these Schwann cells. And these are impinging on the dorsal root entry zone that gets into the spinal cord. And that probably caused the lower back pain. So that was my very initial project and introduction to Alzheimer's disease under the guidance of Leonthal back then more than 20 years ago. And this really resonated with me throughout the rest, or for the next more than 20 years. So I was always interested in neurodegeneration in Alzheimer's disease, and I always had my heart, maybe someday I'll go back to UCSD. But first I did my medical studies and did residency in Erlangen, Germany. So Erlangen, Germany is a very tiny city. It's a college town, 120,000 people when students are around, 70,000 when no students are around. And it's very close to Nuremberg, so it's in Bavaria. And my supervisor and mentor, both clinically and also in research, was back then Jürgen Winkler. He's a movement disorder specialist. And under his guidance, I was allowed to do research and also to see patients in the outpatient clinic later on. Erlangen is not only famous for the university, but if you know about Erlangen, you also should hear about Siemens. So that's a big company, and Healthineers are located in Erlangen. For example, they produce the MRIs. So the MRI here at Radis was, for example, produced in Erlangen, Germany at that time. And then coming from Germany, the one most important thing is beer. So once a year, we have also like a little Oktoberfest in Erlangen. So Oktoberfest is only in Munich, always at the end of September. But Erlangen, I think, has a nicer festival. It's called the Bergkirchweih because it's located at the mountain and it's outside, and you have lots of great food and beer, and it's really nicer than the Oktoberfest, I think. And it's in May, so it's also a different time of the year. As I said before, I was always drawn back to go back into research and also go back to UCSD. So I met Chris Glass 2013 when he was giving a talk in Erlangen. So he was back then introducing epigenetics and genomics and the role of microglia in diseases. And I told Jürgen, this is what I want to do. I really would like to go to Chris, do like a 2 or 3 year postdoc, and then come back. So that was my initial idea, to learn about microglia, the macrophages, and I learned a lot from Chris scientifically, but also personally, and also he made me really grow. We were then able to publish several papers looking at microglia in health context, but then also in Alzheimer's disease to see what are microglia, what is the gene expression of microglia, what are they thinking. Luckily, UCSD San Diego is such a great place. There was not only Chris Glass, but I was also very much influenced by 2 other very important persons. One is Rusty Gage. I actually met Rusty Gage also when I was at the VA and working together with Leon Thal. So that was always influencing me, especially back then, the adult neurogenesis. But then also Rusty was really going into DNA repair, xenotransplantation studies. So I was able and fortunate to also work together with Rusty on DNA repair in neurons, then also work with my friend Simon, who's now in Munich, to work on transplantation of microglia into organoids and this into mouse systems. So Rusty is always, until this day, always influencing my thinking as well. And I was very fortunate also to meet Joe Gleason, who introduced me to somatic mosaicism, something I've had no idea about that was existing actually. So my idea was always of the genome of a cell is the same in every cell of the human body, but actually that's not true. So every cell is a little bit different. So we accumulate little mutations and somatic variants over our time, not only in cancer, but also under healthy conditions. And for example, in the brain, the neurons, they accumulate mutations at increasing age. And also in Alzheimer's disease, there seems to be like an association with increased mutational burden. And this was really how Joe also influenced my thinking. So there are a lot of people that influence thinking, but I would also like to shout out the Rex Scholar program. I didn't know about the Rex Scholars program when I got here, and I actually don't know who introduced me to the Rex Scholar program back then. I forgot, but I'm very thankful because I think it's a great opportunity to network. I think that was the most important thing, to really meet other people across the Mesa. Also was fortunate to go to like the ADRC meeting, and this was really very influential, or to meet then people from outside UCSD, so from Kansas, but also from Columbia and other places as well. So Lunch and Learn series are actually a great place to also meet other people. And also with the Rex Scholar program, I was able to generate preliminary data that actually led to then funding from the Bright Focus Foundation, which I got last year, and also from the Alzheimer's Association, which is very important, especially in these times, to get like non-federal funding and to get the work that's sometimes very expensive to get going. My research program, basically in my lab now, is really focusing on being translational. So I like to work with the human brain or human plasma samples or blood samples, then link it, the mechanism, in mouse models and also in iPSC or INs together with Jerome, back to the patient. And what I started in Chris's lab, what is still one topic of my lab, is really what are the cell type specific mechanisms underlying altered gene expression profiles in aging and neurodegeneration. Basically, I want to understand how is the cell responding to amyloid plaques, to tau pathology. What is inside the cell happening? So for microglia, we know the morphology changes, but also we want to know what is happening inside the cell in microglia, but also in neurons and other cell types like astrocytes and oligodendrocytes as well. So that's one thing, and that's really influenced by Chris. In my lab, also with funding from the REC, I'm now interested also in looking at what is the contribution of DNA damage and also DNA repair mechanisms to altered neuroglial function in Alzheimer's disease. So we know that DNA repair, the activity actually declines over aging. This could then lead to increased mutational burden. So that's another question that we try to answer in my lab. And the third question, a little bit shifting away from the microglia, although we always be in my heart and always, I think, they are most important cell type in the brain. I'm now shifting also the focus to oligodendrocytes. So in context of Alzheimer's disease, we know there's a lot of white matter changes associated with Alzheimer's disease. And I think the oligodendrocytes or oligodendroglia, as the myelinating cells of the brain, they're a little bit underappreciated in the context of Alzheimer's disease pathogenesis. Especially if you think about this, that myelination occurs a lot during your first 2 decades of life, then through your 50s, you have a stable myelination dynamics, and then during aging, actually it declines. So it goes down, and that also correlates with the onset of neurodegenerative diseases. So because it's a rec, I briefly want to just touch about microglia. What are the microglia? Why do I love them so much? So basically, microglia are macrophages of the brain. So they're part of the innate immune system. They respond quickly to pathogens. In the context of Alzheimer's disease, they respond to aggregates like amyloid plaques and so on. They're now 106 years old. They were discovered back then by Rio del Ortega, who did like a series of papers looking at the morphology and also back then already identifying them as not coming from within the brain. So those microglia, they come from the yolk sac and migrate from the yolk sac into the brain. In mice, we know before the blood-brain barrier closes, and then they repopulate the brain, but they don't get any contribution from the periphery. And this is how microglia looked like when he was drawing those microglia back then, more than 100 years ago. So you have this small cell soma, and you have a lot of branches. And what those branches do, they're very active, they're not resting, they look for changes in the environment. So microglia, a very important feature of those cells is they respond to changes in the environment. If they detect something in the environment, they respond by trying to eliminate this pathogen, or they're trying to secrete cytokines, or growth factors for wound healing and so on. And together with my friend Simon, I love this video, so I'll show it again, although probably some of you already saw this video, to show that microglia are very active cells. So here in the middle, you have the microglia. They are labeled as white cells. You don't have to pay attention to the neurons. They're labeled here in red on this side, and this is just the merged picture. But very important when I run the video is that they're really highly active cells, and they really respond to changes in the environment. And you will see after a couple of seconds, there will be a laser injury here in the middle. And then you have to see what's happening to the microglia. So microglia, small cell soma, you have those branches, and they now actively look for change in the environment. Now comes the laser injury, and they migrate those branches into where the lesion occurred. So that's the very essential function of microglia. They respond to changes in the environment. And this is a dramatic change, a laser injury. But imagine for amyloid plaques and so on, there's a similar response. So they migrate also to aggregates and so on. So what are the core functions of microglia? They respond to changes in the environment. They have lots of other functions. For example, they phagocytose synapses, they phagocytose apoptotic cells. That's called efferocytosis. During development, roughly half of our neurons die, and somebody has to take away and degrade those dying neurons. So that's the basic function of microglia during development. They're very important also for synaptic pruning. So they remove connections between nerve cells that are maybe not so stable and so on. That's called synaptic pruning. I mentioned already they're very important for neurogenesis to remove apoptotic cells. They communicate with oligodendrocytes. So they maintain that the myelination is intact and so on. They're basic inflammatory cells, so they belong to the innate immune system. So they respond with cytokines, the classical cytokines that everybody knows, like TNF, interleukin-1 beta, interleukin-6. So these are pro-inflammatory cytokines that can also damage neurons. And they are also very important for tissue repair. So they can secrete growth factors like BDNF, and NGF, for example, to really maintain neuronal health. One problem of the microglia is that they are very difficult to study. So microglia, they're only roughly 5% of all the cell types in the brain are microglia, so you have to get them out to actually study them. So one way to do it is like what we established in the lab in Chris's laboratory with David Gosselin, to really isolate the cells from fresh tissue, that we don't activate the cells. So if you would use enzymes, you, for example, activate those cells and they can have like a different gene expression program and so on. What we did, we used mechanical dissociation at 4 degrees so that we don't activate the microglia. And then we use flow cytometry to actually sort out the cells. So here you can see these are the small population of microglia, and these are all the other cell types. So it's very small, roughly 5% depending on the brain region. This approach always requires fresh cells. So we also knew there are a lot of brains here at the ADRC, and we cannot really use them for isolating the microglia fresh. So we developed a method that actually isolated nuclei from the brain and then did antibody stainings to then get out microglia or astrocytes, oligodendrocytes, neurons, or endothelial cells. And I will go a little bit more over this protocol because it's very essential for my lab, but also for a lot of other papers that we published. And all the papers that I published before or showed before, they're actually based on this nuclear isolation method that I will introduce in a minute. And then if you look for collaboration, I can get out those cells, but then you can do a lot of downstream assays to it. So my lab, we are focusing on single-cell multiomics, so we can look at every single cell and look at their gene expression, at their epigenetic profile. We can do ChIP-seq, so we can look at transcription factor binding. If you have a favorite transcription factor, we can look where it binds in the genome. For the somatic mosaicism, we do whole genome sequencing, so we can really look at every single cell, if there's increased DNA mutational burden, if there are any SNPs, if there are any copy number variations, and so on. One caveat of all those protocols or methods that I showed is you lose spatial information. So when you get out the microglia, you basically lose the context where they were in. So you don't know to what neurons they were talking or to what astrocytes or oligodendrocytes they were talking to. So we're also now doing spatial transcriptomics, where we can then actually look at the communication between microglia and neurons and astrocytes, what you would lose with those isolation methods that I showed before. But for this talk, I thought I'd focus on the nuclear isolation method. So basically, we don't need much brain. So I'm showing here a sagittal cut of a brain. So we usually need roughly 100 milligrams to 200 milligrams of tissue, which we then homogenize. There are several devices that we can use to homogenize. Then we wash. The washing step is very important. And also we do like a sucrose gradient to get rid of the myelin. So the brain contains a lot of myelin. That's basically fat. And that's just disturbing in our downstream assays. So we want to get rid of as much as myelin as possible. So for that, we use sucrose gradient. And then basically, after you have like homogenized and cleaned up the debris, you get like a tiny little pellet of cells. And these are neurons, mainly oligodendrocytes, and then some astrocytes and some microglia and so on. And then we subject them to flow cytometry. So then we can really isolate microglia or the other cell types. And after then performing your RNA-seq, ChIP-seq, single-cell proteomics, or whatever you want to do, we can then sequence those cells. And this is, again, how it looks like when we use our antibodies to then dissect out different cell types from the brain. So basically, you get a lot of noise. You gate on singlets. You get rid of the myelin and so on beforehand. So it's relatively pure. And then here, this is the FAX plot. You can see here a lot of unstained cells or nuclei. And then here, nu N, that's a marker for neurons. So you get a lot of neurons from the isolation protocol, and you get some PU1 positive microglia. So PU1 is a transcription factor that's very important for microglia, and only in the brain microglia have it and other perivascular macrophages. You can also then sort out oligodendrocytes and also astrocytes, which are a little bit tricky. So I personally don't like working that much with astrocytes because they are more tricky to get isolated than, for example, microglia or oligodendrocytes or astrocytes. So we are interested in the transcriptomics and epigenetics. What do I mean? Just a little bit 101 on gene regulation and gene expression. So basically, when a gene is expressed, RNA polymerase, does it work? Does it work and elongates mRNA? You have your promoter beforehand, different transcription factors activating the promoter, thereby increasing messenger RNA. And you have your enhancer region that I'm very interested in, that's very much upstream of the promoter, and that enhances your gene expression. And you have your lineage-determined transcription factors like PU1, and you have signal-determined transcription factors that come in when, for example, the microglia become activated. And you have a lot of enhancers, so promoters, you have in the region of 20,000 promoters, but in the enhancers that regulate gene expression, we have roughly 1 million. And they're very specific for each cell type. So basically on the enhancer that are activated, you know what cell type you're looking at. And we have different methods then to look at the enhancer or the promoter. So one is what I often use is ATAC-seq. So ATAC-seq is a method that looks at open chromatin. So only work can happen where there's open chromatin. When the chromatin is closed, there won't be any gene expression, no transcription factor binds, there's no activity. With ATAC-seq, you can at least say there's open chromatin, something may happen here. But you're not really sure if really something happens only because it's open. And then you have other markers, like, for example, HBK27 acetyl. So here you have your histones that are surrounding the enhancer region. And one marker that shows that you have active gene regulation is HBK27 acetyl. So it's acetylation of histone 3 lysine residue 27. So you can look at this marker, HBK27 acetyl, and say that there's activity going on in this genomic region. So that's your ChIP-seq marker. And just as a reminder, again, at ATAC-seq, open chromatin, you have an enzyme that cuts open chromatin that's shown here. So you have here your histone marks, and that's open chromatin. And when there's open chromatin, this enzyme can cut, and this you can sequence. And then you know you have open chromatin there. And for the ChIP-seq, and this is an example, and maybe it's good because I'm looking at this like almost daily, but I know it's very unfamiliar for a lot of other people. So this is how it looks like when you have open chromatin. So here you have your microglia that are sorted out, neurons, oligodendrocytes, and astrocytes. That's your gene, CX3CR1, for example. That's a microglia-specific gene. And then here you have your promoter in this black box. And you can see here that this region is open. So you only see this open region in the microglia. And then it's closed in neurons, oligodendrocytes, and astrocytes. So you know here basically already on this example that you have a microglia here that has open chromatin at this particular gene. NEFL2 is a neuronal gene, so you would expect it mainly open in neurons. And when you look at this here, so the gene goes from right to left, you see it's not really open in microglia, but you see it's very much open in neurons. And that's called a peak. So here you have open chromatin in neurons. And you can do the same for oligodendrocytes. So here it's only open in oligodendrocytes, and this is only open in astrocytes. You don't know if it's active or not. So for this, you do ChIP-seq. So ChIP-seq is basically chromatin immune precipitation. You use an antibody, in this case, against this HBK27 acetyl that marks active gene regulatory elements. And then you can look if this antibody marks regions that are active. And this is the same here. So microglia, only active in CX3CR1 is only active in microglia. And you can see here in ChIP-seq that's highly active only in microglia. Same for neurons. The gene is only active in neurons and so on. So now you have different measures, gene expression, the ATAC-seq for open chromatin, and ChIP-seq for active gene regulatory elements using HBK27 acetyl. It can get very complicated, so this is like another snapshot of a browser, how it could look like. So here, we basically showed that cell 1 is only active in microglia and not so much in monocytes. So here you have your RNA, you see expression of cell 1 in microglia, but not in cultured microglia, monocyte-derived macrophages, and monocytes. You would expect now that there's open chromatin, and you can see here in microglia there's open chromatin at that gene. You also see a little bit of open chromatin in the cultured microglia, but it's very important to see that using the ChIP-seq, HBK27 acetyl trimethyl or HBK27 acetyl, that you see it mainly in microglia, not so much in monocyte-derived macrophages. This was at the promoter region, and you can identify also enhancer regions. So a lot of upstream of cell 1, you can find this enhancer that's only active in microglia and not in other cell types or macrophages. How do we use this now for Alzheimer's disease? And that's basically why I came to Chris, to really then understand more what are microglia thinking in Alzheimer's disease. Alzheimer's disease is a very multifactorial disease. I think microglia play an important role, But actually, if you look at every other cell type in the brain, you will always find changes. So we'll find changes in astrocytes, oligodendrocytes, and obviously in neurons because they die in the end. You can find also changes in literally every organelle, so lysosomes, mitochondria, everything will be affected in that disease. But I think microglia play a very central role because of their property to induce inflammation. They can then damage neurons. They respond by increased production of amyloid. This can lead to more tau deposition and so on. I think from my perspective, microglia is definitely another target that's worthwhile looking at, not only amyloid and tau, but I think similar to hypertension, where you start with one medication and then you have a broad class of other agents that you use. Alzheimer's disease is probably the same, so it's probably not sufficient to just target amyloid and or tau, but you need also to find other targets that may be worthwhile going after. And I think microglia and inflammation is definitely one avenue to go. Just briefly, microglia, they surround the amyloid plaques. So here in red, you see amyloid plaques, and in brownish, you see those microglia, and you can see the microglia are surrounding the plaques. That could mean they try to digest the plaques and remove the plaques, but can also mean that they're overwhelmed and now they become more inflammatory and then can actually lead to degeneration per se. And that's just another example here from the Salk Institute, which I like, that you really see here in the center the amyloid and then a lot of microglia surrounding the amyloid plaques. There are other indications that microglia play a role. So a lot of the risk genes for Alzheimer's disease, a lot of them are highly expressed in microglia. One example is TREM2. Maybe some of you heard of it. So TREM2 is only expressed on microglia cells that probably interact with amyloid, but also with tau. And if you have a certain mutation in the TREM2 gene that's only expressed in microglia, you can also have an increased risk to develop Alzheimer's disease later on in your life. How I use all my technology then is to really look at what are the transcription factors that drive those different microglia cell states. So basically the hypothesis is always there's a signal, in this case this amyloid plaque, for example, or tau. This binds to a receptor, and then downstream you have an activation of signal cascades. For example, this is a Syk kinase that then leads to changes in the transcription factor usage of the cell. So basically I want to understand what are the transcription factors that make a microglia a good microglia that can be beneficial, or what are the transcription factors that make microglia a more detrimental microglia. So there's this yin and yang of microglia. Are they protective or pathogenic? And I want to understand in both contexts where they're protective, what are the transcription factors that we can use to make them more protective? Or if I know they are now more pathogenic phenotype, what are the transcription factors that make them into a more pathogenic phenotype where they kill then neurons? And basically, then I use ATAC-seq and ChIP-seq to then identify computationally what are the transcription factors, and then I test them also in animal models, how we can influence the binding and switch them or reprogram them into a protective phenotype. I skipped the mouse studies, so I did a lot of mouse studies, and especially when I got to Chris, I was always like, when I look at the mouse and look at the brain of a mouse compared to human, it's so much different. How does it reflect when we study always a human or mouse microglia, what's really happening in a human context? So I was very interested and very intrigued by then studies that came from UC Irvine, from Matthew Burton-Jones, who was always also here at UCSD as a postdoc, when he was actually transplanting human microglia into the mouse brain. You still have the mouse environment, but now you have at least human microglia that you can then study and that may more reflect what's actually happening in the brain that we have. So basically the 5XFAD is an Alzheimer's disease mouse model. I should have said it's more an amyloid mouse model, so it has a lot of overproduction of amyloid, not so much tau, and there's little degeneration. And this mouse is then crossed with an immunodeficient mouse, so you have to get rid of the adaptive immune system, basically, so you don't have mature T cells and B cells because you're implanting human cells. Human cells are then transplanted very early on day 1 or 2 after birth, and then you let them age for 6 months, and then I get out the microglia. So this is how it looks like when the cells become transplanted. So here, so that's a mouse brain. So it looks very much different from a human brain because here you have your huge olfactory bulb. So that's shown here. You have here the hippocampus, you have here the cortex, here's the cerebellum. And this picture is showing the efficiency of the transplantation of human microglia into the mouse brain. So every green dot here is a microglia. So we were very successful in really occupying the olfactory bulb, but also the cortex, the hippocampus, and a little bit also the spinal cord with actually human microglia. And this is higher magnification. So DAPI is a nuclei stain marker. So these are nuclei. And then we have GFP. So the cells were before that labeled with green fluorescent protein. So basically it's a reporter. So you know that these are transplanted cells. And you can see nicely here a lot of transplanted cells in the cortex of this 5XFAD mouse model. And this is another marker, P30 AG062429. That's just a surface marker that's expressed by microglia. And you can see it here. And this is just like a pretty overlay picture where it shows P30 AG062429 and DAPI and also GFP. So human wild-type microglia can really populate the brain of a mouse, which I think is already amazing. And then I was looking what is now the gene expression signature and what is the transcription factors that then drive this phenotype. So basically I was then sorting out microglia. Microglia come in different flavors. So we have homeostatic microglia, we have MHC2 high microglia that we are interested in, but I focus now more on the CD9 high microglia because these are the disease-associated microglia that you see around the plaques. And for me, also a sanity check, does this reflect what we see in the mouse model with the transplanted microglia? Do we see this also, this gene expression signature in human brain tissue? So we were taking a really nice data set generated by the MIT, by Beth Stevens, Molosco, where they were actually isolating microglia from patients with normal hydrocephalus and from Alzheimer's disease, and then we were overlaying our gene expression signature with their data set. So basically what you can see here, the different types of microglia from their data set, and here shown in yellow are our microglia that we were isolating from the mouse model. They were really recapitulating what we see in the human brain tissue. So those clusters here overlap with our mouse model, exactly what we wanted to have. So we think that our model is actually nicely, at least to a good degree, recapitulating what's happening in the human disease context. We were then looking at the competent accessibility landscape. So this is a scatter plot. So basically, I was comparing the homeostatic microglia with the disease-associated microglia. Everything here on the diagonal is the same. So there's no changes in competent accessibility. And you can see here there's a lot of green dots. That is regions of the genome that are open in homeostatic microglia. And you have disease-associated microglia in purple. So here you have gained competent accessibility in the CD9 high cells. And each dot you can then quantify and look what's happening inside. Here's the browser. So this is the CD9 gene, which is the disease-associated microglia marker gene. And you can see here that's the promoter. So it's open in all homeostatic and also in CD9 high. But what you can appreciate here, you have a higher peak in the CD9 high cells. So there's more open chromatin across the cells in those CD9 high cells. You can then, and that's thanks to Chris Glass, because he developed back then with Chris Benner Homer to then look what's happening beneath each peak. So you can then infer what are the transcription factors that bind preferentially in those peaks that you identified. So you compare those 1,776 peaks to background. So you can now say, what are the transcription factors that are preferentially binding here in this new 1,776 regions that are open? And then you get a lot of transcription factors. And that's basically what we look at every day at transcription factors. P1, I mentioned it already, that's a microglia-specific marker. And so it's very comforting to see that this hit came up first because it's a lineage-determined transcription factor. You would always like to see this transcription factors in microglia when you do analysis. And then you see a lot of other transcription factors that are important. So EGR, that's, for example, a family that's very important in immune response. They belong to the AP1 transcription factor family. So when a microglia becomes activated, those transcription factors then also remodel the microglia phenotype. What I was most interested in is actually the MITF-TFE transcription factor family, because this is a family that drives lysosomal biogenesis and lysosomal function. So microglia are very important to phagocytose cells, so they need good lysosomes to then degrade the aggregates and so on that you got. And this is a little bit, there's already a lot known about the MITF transcription factor family in general, but not in the context of Alzheimer's disease, and especially not in microglia. So we were actually the first group that was showing that this MITF transcription factor family plays a role in microglia in the microglia phenotype surrounding the amyloid plaques. So there are different family members, MITF, TFEB, TFE3, and TFEC, and they have a lot of functions. I mentioned they're very important for autophagy and lysosomal function. Need good autophagy and good lysosomal function, and TFEB and MITF, they're very important for lysosomal biogenesis. They also have other functions, lipid metabolism especially. So there's a lot more literature now coming out that lipid metabolism plays a role, especially with ApoE3 and 4 in Alzheimer's disease. That's all bioinformatics. So then we try to knock out MITF, because the idea is then, if this is really that important, if you knock out MITF, then you should see a change in microglia phenotype. So this time we repeated the experiment, but now we also included MITF knockout cells. So we're knocking out the MITF transcription factor and putting it into the cells, and then put it into the animal model. This is again just showing that the transplantation works. So GFP, these are the microglia, they're in the brain of the chimeric mouse system. Amyloglow, that's just a marker for amyloid plaques. So you can see here quantify the amyloid plaques. And Q80 is a microglia marker, or a human marker, not specific microglia marker, just a human marker that we have really human microglia transplanted. And you can see here that those cells that surround the amyloid plaques. So in red, we have our microglia that are surrounding the amyloid plaques. And the amyloid plaques are shown here in white with the amyloid glow. And with the MITF knockout, we also see that the microglia still can surround the amyloid plaques. But what was interesting to see is that we have a less degree of pro-inflammatory cytokines. So microglia secrete TNF, interleukin-1, interleukin-8, which can then harm neurons and lead to neuronal dysfunction. And what we showed is that if you knock out MITF, you see a decrease in those pro-inflammatory cytokines, really validating our findings that MITF plays an important role in Alzheimer's disease microglia phenotypes. Coming to the end, what I'm working on now at the moment is, does this replicate also in human postmortem brain tissue? So that's very important to get resources from the ADRC and others to then look, do we see changes in MITF, TFEB in microglia in human postmortem brain tissue? I like this slide from Dirk Keen. He was giving a talk, I think, 2 or 3 months ago as part of the recruitment for new pathology here. And I really like this mechanism relevance. We can study a lot of things in mouse and also transplant human cells into the mouse, but in the end, you want to go back to the human system. So this is actually what's ongoing now, also to finish the paper, to really look at human postmortem brain samples. So we're trying to validate the MITF-TFE binding in human postmortem AD samples using chip PCR and also PairTag. Just briefly, PairTag is a new method that was developed by Bing Ren here at UCSD that actually can look at transcription and also at the epigenetic markers that I just showed you on a single cell level at the same time in the same cell. So that's pretty new, cool technology that we are now using to look at human postmortem brain samples across normal AD and hopefully also Parkinson's disease, etc. And this is just a browser shot, really that this methodology works pretty well. And then the next step is also then really to find treatments other compounds, small compounds that targets MITF, TFEB, specifically in microglia, and to see if we can then use this system to find some microglia-specific therapies. A lot of data, and I think was pretty fast, but I think the take-home message is just what I wanted to show, or that hopefully gets across, that microglia are really highly plastic cells, so they're not boring cells in the brain, but they do actually a lot and contribute a lot to Alzheimer's disease. If you change the brain environment, those microglia respond, especially to amyloid, and there may be actually promising therapeutic targets in addition to amyloid and also to tau. And what we are also looking at together with Tony Whisker at Stanford is really can we boost lysosomal function in microglia so that they get fitter to then degrade amyloid and other problems in the brain. A lot of people involved. Obviously, this work started with Chris Glass in his lab, got a lot of help, especially with the transcription factor stuff from Nathan. Joy is working on the spatial transcriptomics at the moment. I'm very fortunate to have Sydney. Sydney is my technician, and she took the danger to work with me already 4 years ago, and she's still hanging out. So I'm very happy, and I got now support also because there's a lot of bioinformatics work that we do from Emma. We actually met first time when I was in Chris's, or just started in Chris's lab 10 years ago. I'm very fortunate that she joined my lab this year. And also Suchin, who is great in doing single cell analysis, that really helps. And of course, thanks to Chris, but also to the REC, got a lot of connections to UC Irvine, but also to Harvard Medical School and so on, to then gather data and reanalyze. Thank you so much.